Hello everyone. I welcome you all once again to my series of lectures. So my lecture series I have given a name called as Understanding Pharmaceutical Sciences with Dr. Hariharan. So today's lecture we are going to discuss about the production of rDNA interferon. So I am Dr. Hariharan, Professor of Pharmaceutical Biotechnology. So today's lecture I have divided into three segments. One, a basic understanding about what is meant by an interferon. The second part is about the production of interferon by classical methods, and third is the production of interferons by rDNA methods. So let us go one by one. So first we understand about what is an interferon. So in 1957 there were two scientists, Alec Isaac and Jean Lederman, has identified a chemical from the biological source called as interferons, a potential antiviral agent. So how this works in the body is that when a virus gets infected to a cell, the cell produces a chemical mediator called as interferon. So this chemical mediator gets supplied to the nearby cells, gets attached with the nearby cells, and causes the downregulation of the receptor, and also it causes the inhibition of replication of the viral particle also. Thereby it was started producing an antiviral activity, but this doesn't affect any cellular metabolism of the normal human. So thereafter the interferons has been considered as a potential antiviral agent, because we have very meager antiviral drugs in 1957. So there was a search that a good antiviral agent and a broad spectrum was needed. So let us understand the importance of the interferon. So importance of the interferon is a biochemical mediator. So it is a perfect antiviral agent because it is produced naturally in the body. So we can administer like a thyroxine, we treat an hypothyroidism. So we can supply this drug, that is the interferon, from outside for work as an antiviral agent. Because it doesn't going to produce any toxicity. Anyhow, it's antitoxic because it's non-toxic in nature has been identified, and it was seen in the lab in the cultured cells and animals that it was well tolerated because it is naturally in nature. And one more major findings, importance of this one is that its antiviral activity is broad spectrum in nature, not like the immunoglobulins. As you can see, if you see an immunoglobulins, they are very specific in nature and it binds with a particular epitope and destroys it, whereas the functioning of this interferon is it's active against a wide range of viruses. So it's a broad antiviral agent. So apart from its antiviral and its specificity in action, then there are other properties also contributed its antiviral activity. If you see the various properties possessed by this interferon is that first, it inhibits the cell growth of the viral infected cell. One. Second, it modulates the immune system. As I have already told to you, the interferon gets released by the infected cell and binds with the nearby cell and causes the downregulation of the receptor, which facilitates the binding of the virus. And the third is that it has an ability to inhibit the viral replication. So all these properties contributed to the interferon to be potentially used as an drug of choice. Then let us consider then different interferons started discovering upon. Then in 1980s the Committee of Interferon Nomenclature come up with some suggestion and some abbreviation, and they stated that any compound or any chemical mediator to be qualified as an interferon should be protein in nature, and moreover it should accept an antiviral property non-specific in nature to the virus. That means at least it should be homologously acting on a particular group of the virus, and it should not have an influence over the metabolic process of the humans of its protein synthesis or RNA replication, but it has an impact in the replication of the virus. So based on this, the different compounds started has been identified and be considered as an interferons. So they have given an abbreviation to the interferons like capital letter IFN. So along with the species of origin, for example, a human interferon has been designated as HulFN. So based on this, there are various types of interferons are started identified in humans. So if you see, there are majorly three types of human interferons has been recognized at present, that is called as alpha, beta and gamma. All are protein in nature, but they are antigenically different in nature. But if you see, the naturally occurring one is the beta and gamma, whereas alpha was little bit glycosylated in nature. So later on we identified different forms of interferons. Then there was very much studies has been going on about the characteristics of these interferons. So if you see, the characteristics of these interferons is from the source has been given name and has been identified. So the human interferon alpha is also called as type 1 interferon, called as leukocyte interferon, because it has been seen in leukocytes of the blood cells. So similarly, interferon beta is called as type 1 fibroblast interferon because it was rich in the fibroblast cells. And finally, interferon gamma is called as type 2 or immune interferons. So all these interferons are more or less stable in blood pH, but once we want to use as a drug, it has to see that the stability in the acidic pH. The interferon alpha and interferon beta both are very much stable in acid, that is around pH 2, whereas interferon gamma was an acid labile. And moreover, if you see the molecular weight of all the interferons, it is more or less near to about 20,000 Dalton. That is, it's a single monomer in nature. That means a single polypeptide chain of amino acids. So the number of amino acid present also little bit differ, whereas there was a similarity exists between alpha and beta. If you see, the human interferon alpha and beta composed of 165 to 166 amino acids, whereas interferon gamma has having 146 amino acids. So not only its number of amino acids also differ. If a little bit gets into the structure of interferon alpha, beta and gamma, because the amino acid sequence of alpha and beta are similar in nature, and moreover 165 to 166 number in nature, both are also structurally more or less similar in nature, alpha and beta, whereas the interferon gamma was different in nature. So because of the structural similarity of alpha and beta, they also binds to only the same receptors, whereas the interferon gamma binds with different receptors. So these apart from this, there were many studies, gene cloning studies, to classify whether apart from alpha and beta there are subclass exist or not. It has been identified that interferon alpha has nearly 12 subclasses, whereas beta have 2 subclasses and gamma exists as one. So apart from this, while we go for the production, we have to understand the genetic sequence, especially for the rDNA production. So it has been identified that the genes of interferon and alpha and beta contains only exon sequence. They doesn't have any intron sequence. As you know, there are any DNA molecule carries 2 sequences. One is called exon, another is called intron. Exons are coding sequence which are going to produce the protein, whereas introns are non-coding sequence which generally doesn't produce the proteins or doesn't produce any functional proteins. So the interferon alpha and beta doesn't contain any intron sequence. It generally has an exon sequence, so it can be easily manufactured by rDNA technology. Take the DNA sequence, clone it in a vector, then insert it in a bacteria or yeast, it produces in large quantities. Whereas the interferon gamma becomes a little bit difficult in nature because it has 3 intron sequence. So how we are going to produce by rDNA technology of interferon gamma becomes a questionable. So since we have taken interferon gamma production rDNA as an example for today's lecture, so we will study later on how it has been produced. So the genes which are generally controlling the production of this interferon alpha, beta and gamma are generally present at different location of chromosomes. So alpha and beta was present in chromosome 9 of the humans, and chromosome 12 controls the interferon gamma. So apart from this, the interferons are highly biologically active in nature. If you see, it has a specificity around 10 to the power 8 units per milligram of protein. So it is highly specific in nature. So let us understand how the production of interferons has been happening. So if you want to produce interferons, what you have to do is, there were two production steps. One is the classical step and second is the rDNA step. First we understand the classical methods, how the production has been happened. So in classical methods, once we want to produce the interferons, means first it should be stimulated, it should be induced. The cells should be induced to produce interferons. So a wide variety of substance has been used to stimulate the production of interferons. So naturally, if you want to produce interferons, what you have to do is you have to stimulate the cell by infecting with virus. So generally many animal viruses has been used for the production of the interferons. So let us see the viruses of different type, but the most possible one we see is the perimyxoviruses. Example, Sendai viruses and the arboviruses were more efficient in nature of stimulating the production of the interferon. So apart from this, we have chemicals also to produce the interferons. The time interval between the application of the inducer and the production of the interferon varies. So if you see that generally the chemicals poly, IC and certain bacteria produce 2 to 8 hours, they have yearly inducers, but the amount of production were less. Whereas the viruses have 6 to 16 hours for induction, but it produces a large quantities of interferons. So different types of interferons as in human alpha, beta and gamma are obtained from different locations. So human interferon alpha is obtained from the buffy coat of human blood, which has been induced by SEDI virus. So that's why we call this as a leukocyte interferon. So similarly, interferon beta is a fibroblast interferon because it is obtained by the induction of poly IC into the human fibroblast. So that produces the interferon beta. Whereas the mixtures, mainly interferon alpha and beta, can also be produced because both the mixture alpha and beta can be obtained by infecting the virus to the lymphoblastoid cells. So lymphoblastoid cells once get infected with a virus, it also get induced and causes the production of a mixture of interferon alpha and beta both together. Whereas interferon gamma is produced only by the T lymphocytes in response to stimulation of the virus or any of the mutagens. So this is how naturally we were producing from various animal sources the interferons, but this has leads to some limitations because the amount of interferons alpha and gamma produced by these methods are very less because of the availability of the leukocyte. If the leukocytes are less in quantity, generally we have less amount in nature. So whereas similarly the interferon beta was also very less in quantity because it is based on the surface area of the glass which are exposed to the growth of the fibroblast. So the yield was very less by this classical method. So we need an rDNA technology to produce interferons in large quantities. So the basic principle, if you see the production of interferons in rDNA technology, is that we can produce the interferons from both prokaryotes and eukaryotes, that is from an E.coli as well as from a yeast cell. So that we have seen a successful expression of E.coli both in the genes of human leukocyte for interferon alpha and interferon beta in fibroblast. So these yields are generally very higher, but the very higher one we can get it from it is higher when compared to the classical method, but still we got a higher yield from an eukaryotes, especially from the yeast cells. So there was a little bit theory behind this rDNA technology since we have studied the various steps of gene cloning. So there was how the interferon alpha and beta has been that both doesn't contain any intron sequence, so the gene can be spliced and can be integrated easily. Whereas the issues occurs with the interferon gamma, as you already told that interferon gamma carries genes along with 3 intron sequence. So splicing 3 intron sequence very precisely and joining the remaining sequence and getting the interferons is a difficult task. So we have planned to produce the interferons in little bit different way. So first what I have done is they have taken an interferon messenger RNA it has been extracted from the cell, then it has been converted to a DNA by a complementary DNA copy, that is cDNA was made. Later on, this cDNA has been attached with some short nucleotide sequence. So what are all the short nucleotide sequence we have attached? Generally includes, one is a promoter sequence. Why we need a promoter? Because we need the expression of the cDNA. So cDNA is simply a conversion of mRNA into a DNA, whereas the DNA sequence carries already the promoter sequence, attachment of ribosome site, all will facilitate the expression of the gene very easily, whereas cDNA expressions can be done only by attachment of these sequences. So what we are doing is, after getting cDNA, we are attaching this sequence in the front, like the promoter sequence. The promoter sequence we are attaching to work with a certain operon system. In this case, we add the gene of trypoperon, that is, it has a specific separate of tryptophan. Once I add the tryptophan into the media, what happens is this trypoperon gets stimulated and it causes synthesizing the expression of the interferon. And second is the ribosome binding site, because the sequence should allow the ribosome to bind, then only the translation can happen. And third, it has a linker molecule, so I can attach the gene for the translation. So these things has been included. So the linker molecule facilitates the restriction, then we can attach it to the necessary vector. So after that, what we will do is, first we take the cDNA, attach this promoter, we amplify with a PCR, then this we attach with a linker carrying, then we produce in a large quantity. Then this should be attached into a vector. So the vector generally carries an, as we studied while in vector, it carries some antibiotic resistant markers, so we can get the cloned vectors and thereby we can produce in large quantity. So if you see the production process is that generally the DNA, cDNA sequence, for human leukocyte interferon is very easy. We can clone directly into the yeast alcohol dehydrogenase gene in the Saccharomyces cerevisiae and produce in large quantities. So I have already told the production of alpha beta is different from gamma. Gamma only we need the cDNA, whereas alpha beta we doesn't need any cDNA because itself it's a gene. So that gene can be directly inserted into a vector, especially at the precise site where it can express it. In this case, we are inserting into an yeast cell, more precisely the plasmid vector of the yeast alcohol dehydrogenase gene, and thereby expressing it. So it produces a large quantity. Similarly, we can do the same thing in the E. coli also. It doesn't have a matters. So later on, if you see the human interferon gamma gene, so I have explained with a picture for you. So if you see that in this thing, what we are doing is that we are taking an mRNA of the interferon gamma. If you want alpha also you can do it. So we have taken an interferon alpha also in this case. So we have taken an mRNA sequence, then we run a PCR. So in the PCR we are converting this mRNA into a cDNA. So we produce a large quantities of cDNA. Then to this what we are doing is we are adding a sequence for the desired expression. Like what we are adding? We are adding the promoter sequence, we are adding the ribosomal sequence. So ribosome binding site and promoter sequence both are linked, as you can see from the figure. Then this has been cloned into the vector of our choice. In this case I have taken a Rosetta 2DE3 vector which can easily express in an yeast cell. This Rosetta 2 is an expression vector. So it has been cloned, inserted into a host cell, generally an yeast. So once it is inserted, what happens is that it has been identified by its marker, then it is cultivated in a fermenter. So once I cultivate in a fermenter, it is produced in large quantity, then I calculate it for its downstream processing. So once I downstream it, I have to take care how I can isolate the interferon in pure form. So what happens is, why we prefer the yeast is, if I use this in an E. coli, it produces some bacterial proteins which becomes very difficult in purification process. So moreover, the second problem in the downstreaming is the chemical purification. So the chemical purification of interferons has been very difficult because it leads to small amount of isolation of interferon or sometimes loss in its activity. So nowadays the purification has been done by using monoclonal antibody affinity chromatography, which gives an absolute purification of the interferon. So it is up to the technologist to use a proper downstreaming process for the isolation of the interferons. So I will thank you very much for your continuous listening to my lectures. So interferon alpha, beta or gamma has been produced in large quantities nowadays as an antiviral agent. So thank you very much. So I prefer that you will continually